Hello, everyone. I'm going to talk about my research in the topic of optimized delivery of Mcl-1 and survivin siRNA combination in a breast cancer cell model in vitro. The breast cancer ranked the first in cancer incidence among women with more than 2 million new breast cancer cases in 2020, with an increasing trend in majority of the cases. Development of breast cancer involve estrogen, progesterone, and epidermal growth factor receptor, while another sizable fraction, 10 to 15%, involve triple negative cases. Trend in development of novel cancer therapeutic have shifted from conventional to more specific targeted method. Conventional target treatment strategy focus on the use of monoclonal antibody. The targeting oncogene in malignant cell by inhibiting oncoprotein synthesis is a relatively new approach for molecular targeting. Small interfering RNA or siRNA is a promising intervention in this regard since it can overcome the limitation of small molecular pharmacological agent by targeting previously undruggable cancer driver at will. siRNA implement its gene silencing activity by incorporating itself into the RISC RNA-induced silencing complex, which then bind to target messenger RNA or mRNA in order to degrade it, resulting in attenuated protein synthesis. The siRNA complex preparation. The trans booster additive was added to the siRNA that was dissolved in the rPMI at 1:1 siRNA to additive ratio weight to weight with a final siRNA concentration of 40 or 80 nanomolar in the tissue culture medium. The desired transfiction reagent was then added at a 1:5 or 1:10 weight by weight transfiction reagent to siRNA ratio. And the mixture was then incubated for 30 minutes at room temperature to allow for optimal complexation. Result and discussion. The increased cell killing was observed with increased siRNA to polymer ratio. At 80 nanomolar siRNA concentration for the Mcl-1 siRNA in figure 3a. No effect for CDC30 or CDC20 and HSP90 siRNA treatment were observed. At 80 nanomolar siRNA concentration and 1 to 7.5 to 1 siRNA to polymer to additive, the cell viability was the lowest at 70% with Mcl-1, figure 3b. Treatment with siRNA give insignificant, lower than 10% cell death under this condition. By increasing the siRNA to polymer weight ratio from 1 to 7.5 to 1 to 10, we obtain a higher rate of cell killing in figure 3c. The inhibition of cell growth was approved to 45% as compared to the non-treatment group. Lipofectamine appeared to have a higher toxicity than Pamphix, despite the lower siRNA polymer ratio. Figure 3d. When the number of cells per ml was varied from 10,000 to 40,000 cells per ml, the non-specific toxicity of siRNA was decreased. The cell viability was increased from 45 to 80%, while the efficacy of Mcl-1 siRNA at siRNA to additive ratio was increased. The additive ratio will vary at 0 to 2, as can see in figure 4b and figure 4c. optimal siRNA to additive ratio was 1:1. The highest cell death was detected at day 6 after transfection at 1:10:1 with 80 nanomolar of Mcl-1 siRNA from the figure 4E. Cell viability was approximately 35% on day 6 and approximately 50 on day 3. Figure 5A and B, according to the result of the experiment, the combination of siRNA has very low cell viability of around 20% and 30%, and is statistically significant when compared to the single surviving siRNA treated group in MDA436 cell and MDA231, respectively. Figure 6A and B at 60 and 80 nanomolar dose, the fold change of MCL siRNA treated group was around 0.4 and 0.3 respectively. The fold change in 1:10:1 and 1:10:1 groups was about 0.4 and 0.26 respectively. Give the remaining percentage of the target mRNA to make it correspond to the cell viability. Conclusion. The use of additive result in improved cellular uptake of siRNA and dramatically increase cell death. This study optimal condition were 1:10:1 siRNA to polymer to additive weight ratio, 80 nanomolar siRNA concentration, and 6 days transfection with PAMFIC transfection reagent, which result in achieved 30% cell viability compared to the non-treatment group. The combination of Mcl-1 and survivin siRNA show the more powerful than the single Mcl-1 and survivin siRNA. The cell viability of the combination siRNA is approximately 20% with 3 days of transfection. In the following phase, this study needs to be studied in animal model to confirm the effects of the combination of Mcl-1 and survivin siRNA additive polyplex using PAMFIC transfection reagent. Thank you for your attention.